mechanism for its RNA, right? It has its a repair mechanism for its RNA. So its mutation rate is not as fast as you would see with HIV, for instance, or with the other RNA viruses. So initially, when studies on vaccines and vaccine development started, we thought this was actually quite a big benefit because you would have a vaccine that that would last for quite a bigger time. And time has proven us wrong. It's quite a very clever bug, this one. It also has the envelope, an envelope around it. If you remember from your basic microbiology at medical school, viruses are either naked or they've got a coat. The coat is the envelope. It's a lipid envelope. And what is the importance of this envelope? The importance of this envelope actually it tells us that this virus is vulnerable to desiccation and disinfectants. So it's a virus that cannot persist for a very long time in the environment. And then two, it's highly susceptible to your alcohol disinfectants and your normal disinfectants. And then it's got a membrane cover and the now notorious spike protein. Now the spike protein is the protein that it uses to attach to the receptors on the host. And the commonly used receptor is the ACE2 receptor. That's the angiotensin converting enzyme 2 receptor. And this enzyme is promiscuously available throughout the body. It's in your GIT tract, it's in your kidneys, it's in your lungs, it's in your nasopharynx. And that's the disaster with this virus. It's got a large sample space of receptor field to choose from. From an immunological point of view, what do we know, right? Earlier on in the outbreak, they took a lot of epitopes that had been collected based on the earlier SARS outbreaks, that is the MERS outbreak and the SARS in 2022. And so when you took these pools and you tested them for CD4 and CD8 responses amongst patients that had recovered from COVID, it was noted that 100% of these patients had epitopes, had a CD4 response. So they had IgG antibodies, 100% of them against the spike protein, and then IgA, which is quite important for your mucosal immunity, against spike protein 2. And this is what gave the direction for people in vaccine development to start targeting the spike protein. Not only that, but there were also T-cell responses, right? And the T-cell responses were actually much more widespread. They were against the nucleocapsid, they were against the membrane protein, and they were against the spike protein as well. So there is a CD8 factor that comes into the immune response to SARS-CoV. And then further testing in non-exposed individuals actually detected these T-cell responses as well. Now, this can be associated with the coronaviruses that we get, our GIT infections, those ones that will cause you a mild diarrhea, and those coronaviruses that are associated with the common cold. So the clinical impact or the clinical benefit from this cross reactivity is still unknown at this stage, and it's still work in progress. But then still we've got challenges from this knowledge of immunology, because we still don't know much about the correlates to protection. What are the correlates that we can use to say, for instance, the vaccine protects you against infection, acquiring the primary infection, or the IgA response that allows you to deal with the virus at your mucosal level, and then you cannot spread it, you cannot transmit it to others. And what are the correlates that we can use to determine disease progression? Or what can we use to predict what that subset of patients that subsequently end up hospitalized or they die? Now, vaccinology has moved in the last in the last 40 years, and particularly since the 1980s, when we had the hepatitis B vaccine, which was the first recombinant vaccine. So since 1986 thereabouts and to date, you can see the technology has moved, right? It's becoming more towards vaccines that are based on recombinant proteins, that are based on molecular modification, that is DNA or your RNA. And you've also had development of adjuvants in the process. What is an adjuvant? An adjuvant is an accessory to the antigen that allows the immune response to shoot several times higher. And what do we see? How have we used this for COVID-19 vaccines? You can now see that we've got about 63 vaccines now that are in clinical development, and even more that are still in the preclinical stage. You've got about 179 vaccines that are in the preclinical stage. And most of these vaccines are using the new technologies, right? So these protein subunits, the recombinant subunits, that's 32% of the vaccines coming in. And the viral vectors, the non-replicating viral vectors, the adenovirus, for instance, it's about 16%, and the DNA in the RNA viruses as well. Now, just a quick note on this new technology. The major vaccines that have come in have utilized the mRNA approach and the non-replicating virus vector approach. What happens with mRNA? Over time, people have learned to preserve mRNA. It degrades easily, it gets contaminated easily. So technology has allowed it to you get mRNA from a baculovirus or another virus, for another virus platform, harvest this mRNA and put it in a transport mechanism, which is usually a lipid nanoprotein. And you inject this into the host, right? What the mRNA gets delivered to the host ribosomes. It's all outside the nucleus in the cell cytoplasm. And then it is transcribed, it is translated into proteins. And these proteins are the spike protein that elicits an immune response. Then you develop neutralizing antibodies. It's quite simple. The other bit is the adenovirus, where you get an a harmless, not harmless really, the common circulating adenoviruses, usually the non-pathogenic adenovirus serotype 26 or 20 or 5. And in it, you inject the viral DNA, right? The part of the DNA that codes for the spike protein gets incorporated into the adenovirus. And then the adenovirus is then used as a transport media. It becomes the vaccine. You inject it into the host. The host takes this non-replicating virus, and the virus does its infection process, gets into the nucleus, into the host DNA, and you get transcription and translating into mRNA into the viral protein. One of the side effects which become very important with the viral vectored infections, with the viral vectored vaccines, is already the host might have an immune response to the adenovirus, right? So there's a potential that you will naturally neutralize this vaccine. So that's why you need to have a booster doses for this. What is currently available? Right. What do we have? I'm touching on the vaccines that we have here in looking at basically here in South Africa. This one out of the post. Hello, is somebody?
Hello. Okay. Let me try to mute this person.
Okay, can you people go on mute? So what is the first one out of the gate was the Pfizer vaccine. The Pfizer vaccine is using mRNA technology. And in the clinical efficacy trials, if you looked at clinical infection, the prevention of mild to moderate disease, it was 95% efficacious. and its efficacy against hospitalizations, protecting those that got infected, got disease, but did not progress to severe disease needing hospitalization was about 90%. How do you administer this vaccine? It's 2 doses, 21 days apart. What's the big drawback with this vaccine is it requires -70 storage, right? You store it at -70, you transport it at -70, but once you take it out for vaccination, it is stable at 2 to 8 degrees in ordinary fridge temperature for about 120 hours. The second one that came out in mRNA based was the Moderna, with same efficacy rates and same protections against hospitalization. In the interest of time, I'll actually then touch on 2, I think 3 that are quite interesting on this slide. There's then the AstraZeneca, which has caused a lot of debate. It's a viral vector vaccine. That is, it's based on the adenovirus. Right. Now, in clinical trials, there was varied protection, right? If you looked at it, depending on geographic region and depending on the dose. When they started the trial, they were using a standard dose for first inoculation and the second inoculation. And the first inoculation was noted to have lower efficacy at 67%. Than the lower dose, which gave efficacy at 90%. The reason being what I explained earlier on the natural neutralization of the vaccine by pre-existing antibodies, right? If you give it at higher dose. Right, so this AstraZeneca vaccine comes in 2 doses, and it was going to have a very big global impact because it is stable at 2 to 8 degrees. You can store it at 2 to 8 degrees, you can transport it to 8 degrees and vaccinate at that. The other vaccine, a viral vectored vaccine, is the Johnson & Johnson vaccine. And this has been very interesting because if you look at a protection from infection in the clinical trials from the South African centers, it only had protection of about 57% if you looked at acquisition of infection. But it was efficacious in protection against hospitalization. That is, in protection against severe disease with 85%. And this was across all the clinical trial centers, both in South Africa, in Europe, Brazil, and the United States. And the beauty of the Johnson & Johnson, it's a single dose, and it also stores at the same temperatures as the AstraZeneca. Sputnik is interesting for other scientific reasons because we are looking at heterologous adenovirus injections here. And Sinopharm is an inactivated virus vaccine, and it's from China. From the clinical trials in China yet to be published, you see efficacy rates ranging between 50 to 70%, and it's also a 2-dose vaccine. Right. So if you are going to then choose a vaccine, what are the considerations that you want to pay attention to? It's the safety, right? It's a vaccination, you are vaccinating healthy people. So safety is very paramount. It's the efficacy, and particularly in the populations the high-risk populations that we are talking about, the cost, the transport and storage, and the dose regimen. And what we have learned recently with COVID is you need very good surveillance programs. Right, surveillance programs to pick up changes in your strains that are circulating, emergence of variants that can affect vaccine efficacy. Because vaccines have a core business, and their core business is to survive, and to do that they mutate, and they also recombine, and that's how we get new viruses and expanded host ranges for viruses. And what have we learned with the COVID variants? Right, first, right early in the beginning, we had the D614 mutation that happened in China in March 20. Right, what the impact of this change in the virus was, it increased transmissibility. It became more efficient in binding, right, and then became more transmissible. And it is this mutation that caused it to become a global phenomenon, to be so widespread. Then within a short time, in September 2020, there was the 4531, right? And then that mutation was in Denmark and the Netherlands, right? We saw a change here where the virus actually became adept at infecting the ACE2 receptors in mink, right? So you now had human to mink transmission, mink to mink, and mink to human. And the implication of this from a public health point of view is you are creating an animal reservoir. So whatever you do to this virus, as long as there is a reservoir in the animal, it will jump back into humans. So it has resulted in massive culls on mink farms in Denmark and in the Netherlands. Then we had lineage B117 from the UK. Now, this mutation also, this lineage has a number of mutations. That's why we call it a lineage. About 23 of them, and 17 of them are actually the important ones. And they have resulted in spreading 56% times faster than the normal variants. And it has higher affinity for the receptors. And what we think is it's a reflection of the magnitude of community spread and a chronic infection, giving the virus an opportunity to select for survival. And then we've got the South African variant, B351. Now, it detected during this current second wave, and it was quite fortuitous because it was at the same time that the clinical trials for the AstraZeneca vaccine, the Novavax vaccine, and the JJ vaccines were happening in South Africa. And it has become the 80% of the circulating strains in South Africa with high viral loads in the specimens. And what it means is you've got higher transmission rates. And it has got two worrying mutations, right? And these mutations have been picked up in the UK lineage as well. That is 417N and 484. And these two mutations allow the vaccine to escape immunity, right? So it will escape natural immunity. That means we're now seeing patients that were infected in the first wave coming in sick in the second wave. And there is now evidence of this variant affecting viral efficacy. And like I was saying, the impacts of variants developing, they tend to spread faster, they become more transmissible. We don't have evidence of increased mortality or more severe pathogenesis in the new variant patients. But what's quite interesting, why I put up this slide, is the politics that's associated with these bugs. The British started saying it was the South Africans who came up with this, and then the South Africans had to issue a media statement to say, no, we are not responsible for this. It's actually in the behavior of the virus. And what's quite nice is we noticed this variant when the trials were actually happening. I put this slide because of what the Africa CDC has said, that about the AstraZeneca vaccine, that you can proceed to use it if you don't have a variant that's found in South Africa. But how many countries have got surveillance mechanisms to know what are the circulating variants in their communities? I'll have to go a bit fast in the interest of time, but are viral vector vaccines effective against the variant that we have seen here? In South Africa, we know AstraZeneca vaccine efficacy goes down up to 22% when you are confronted with this variant. The Johnson & Johnson vaccine seems to do better. The efficacy goes down to 57% and 72% in the US, but we'll see the US will revise downwards as they get these variants in their population now. Now, the Johnson & Johnson vaccine seems to protect against hospitalization in 80% of these patients that are infected with the new variant. In the laboratory, if you do neutralization assays, you will see that the antibodies go down in the AstraZeneca vaccine by about 86-fold. So it's actually quite severely affected by this new variant. Now, the protein subunit vaccines, one of the vaccines that is in the pipeline is the Novavax vaccine, and that was on clinical trial here recently. And during the trials, when we were having this new variant, overall efficacy down, bomb, 49%. Right. Now, if we look across all the vaccines and from laboratory studies, we don't have much studies on the Johnson & Johnson in the laboratory, but we know the Pfizer and the Moderna, the Pfizer vaccine, the mRNA vaccine, neutralization antibodies go down threefold. There is no data yet when it comes to the clinical efficacy. We know Moderna goes down 6, AstraZeneca up to 86. So it actually falls out when when you do laboratory studies. Clinically, what does that mean? Right? Johnson & Johnson is still okay because in terms of efficacy against severe disease and against hospitalization, we still have something in the arsenal. We don't have clinical data for Moderna and Pfizer. AstraZeneca, clinically in mild and moderate disease, 20%, 22% efficacy. But what we don't know is the level of protection in severe disease and in disease that requires hospitalization. That's why the rollout program was stopped, and it's now been introduced again in a stepped up kind of approach to see whether it has an impact in severe or hospitalized disease. We still need to move further with Novavax vaccine. Its efficacy sitting at 49%. We now need to elucidate what's that impact going to be in severe and in hospitalizations. So what we have in summary and what we know about the vaccine against what is happening in the strain here in South Africa is there is laboratory evidence that there is a reduction in vaccine-induced antibodies. And that reduction varies from minimal with the mRNA vaccines to quite substantial, particularly if you look at the AstraZeneca vaccine. Right. Now, we've got clinical trial evidence now of reduced efficacy in mild to moderate disease for AstraZeneca vaccine, 22%, Novavax at 49%, and the JJ at 57%. But we still need data for the mRNA, and we still data. Right. And we still need data against hospitalization. Now I'm on my final slide, and as a clinical micro, I won't do this justice without talking about diagnostic testing, right? What is the impact of genetic variants on diagnostic testing? Currently, it seems to be minimal, but if your laboratory systems are using the targets based on the S gene, be very careful. You need now regular monitoring to see the performance of your laboratory tests against these variants, because you will end up with a lot of false negatives, right? You will have people with clinical disease with the laboratory saying, no, there is no evidence of COVID. So in conclusion, really, we will see these variants continue to emerge as we get widespread community infection. The impact of these variants on antigenicity, vaccines, and the clinic is still unpredictable. So you will need very active molecular surveillance so that you know what strains are circulating in your population. And the vaccine manufacturing approaches need to be agile enough to be able to change the genetic formulations of the vaccines to cover the new or the emerging variants in the same way that you do with the flu vaccine. And the biggest lesson that has come from the South African vaccine program is you need actually a phased approach to vaccine introduction, right? Particularly where you've got variants that are becoming predominant, right? You want to know efficacy at different time points, at infection, for severe disease, for hospitalization, and ultimately, is it effective in preventing death? Thank you, and my apologies for taking a bit longer than I should have.
Thank you, Becky. Thank you very much for the very, very impressive presentation, very scientific. I think we would straight away go into questions so that at least we try to preserve some time. I think from the chats, Dr. Chirisa, Macron Chirisa, you have a question. Please, can you go ahead and ask the question?
Thank you very much, colleagues, for this very interesting presentation. I have two questions. The first one is, I think you mentioned that the vaccine manufacturing approaches need to be agile enough to be able to change the
very wonderful webinar, and thanks, Becky, for such an insightful presentation. My question was just on I was focusing, on when you're looking at the vaccines in terms of what we are looking at introducing in our own country in the next few weeks. That's the Sinopharm and the Sputnik vaccine, but specifically the Sinopharm, which is an attenuated virus. And I was just wondering what impact that would have in terms of.
John.
Oh, okay. Let me start again. What about now? Can you hear me, Dr. Maulana?
Hello, is everyone hearing
Yes.
what Majoni?
Yes, we can
hear you. We can hear you.
Okay, fine.
Yes,
Okay.
we can hear you.
Okay. Okay. So my question was, being an attenuated virus, what is the impact of mutations? What is the likely impact of mutations, given that it's a particular, I'm not so sure about the science of an attenuated virus, but what do you suppose would be the impact in the event that we've got mutations in the community?
We can't hear anyone.
Yeah, thanks, Mark John, for that question. The live attenuated, this is a whole live virus that is weakened so that it does not cause disease. And this is the virus that the Chinese have been working on, and they've put on clinical trials in China. And I think it's now getting global acceptance. Now, the mutations that have been quite significant have been for the new technologies, right? Because they are based on the spike protein. So that will affect the efficacy of the spike protein. If you look at the lab studies with the new variants, right, the neutralizing antibodies with the Sinopharm vaccine have remained kind of, there's been very minimal impact on it. That's why the smile on that slide. Now, another benefit that you have with the live attenuated vaccine is then the body responds to other epitopes other than the epitopes on the spike protein, right? You now have immune responses or T-cell mediated responses to the M protein, to the N protein, and to other sites on the virus that were not targeted by these other vaccines. But still, you will need to monitor the impact of a live attenuated vaccine in immunocompromised patients, right? You know that they might be vaccine-induced disease. I don't know whether I've answered you there.
Thank you very much, Becky. Please, if there are any questions, go ahead. One or two more questions, go ahead and ask, so we can move on.
Hello, Dr. Mwale. My apologies. I can't see where to raise my hand there. But this is Nyarazo, Dr. Mgodi. Thank you very much, Dr. Magazi, for your presentation. You talked about neutralization. I just wanted to find out, do we, at the moment, do we have any neutralization marker that we can, an assay that we can use as a marker to see how a population is going to respond, or how effective a certain vaccine is going to be in a certain population? So I just wanted to find out if we do have any potential neutralization assays that we can use so that we are well informed. I'm talking from my background of HIV prevention. We are working on certain markers similar to that. So I just wanted to find out, in COVID-19, do we have specific assays or any potential assays? Thank you.
Thanks, Nyarazo. To the best of my knowledge, not at this stage. And like I mentioned earlier on in the presentation, in that basic immunology slide, is we are still at sea here when it comes to what are the markers of protection, right? What are the markers that we need to be looking at for protection against infection, against transmission, against disease severity, and against death.
Okay, thank you.
Hello.
Okay, thank you. Are there any more questions, guys?
Yes, Dr. Mari wants to talk.
Hello, Dr. Mane.
Hello?
It's Dr. Mari from Australia.
Oh, okay, please go ahead, Dr. Mari. Thank you very much. Go ahead and ask, please.
Oh, thank you so much. Thank you to the organizers of the webinar, and thank you to Dr. Magazi for the nice presentation. I've just joined it midway. My question is on the efficacy of the new vaccines. I can see your slide, which is on at the moment, which is puts Johnson & Johnson, the single dose, quite reasonably protected from the new variants. When you look at a country trying to buy one vaccine for the nation, what would you suggest is the best way to look at the different vaccines coming in, besides the storage, transportation challenges? And second question is, with already populations infected, how efficacious are these vaccines going to be vis-a-vis people who have already been infected and they've got some antibodies?
Thanks, Dr. Mari, for that. If we are looking at choice for a vaccine, there were those five considerations that I had put on in an earlier slide. To let me find right. These would be the 5 considerations that we would actually want to look at. Right, and safety paramount, because this is you are giving this to quite healthy people. And I must add here that what has been noted with the mRNA vaccines is you get more reactions to the vaccine than you would with other normal vaccines, and that's mainly driven by that lipid that you use as a carrier for the mRNA. So, but these are not severe enough to stop vaccinating with the vaccine. You will need to be very cautious when you, I see HTO has got his hand up, and he will tell us from the UK experience. You've got anaphylaxis that has been severe in a couple of cases. That's why the recommendation is to stay for about 30 minutes after getting it. And efficacy, and then there's the issue of cost, transport, dose regimen, and you develop a surveillance program. How should we introduce them? It's a question that has seized the South Africans this side, and what they have come up with is a phased-in introduction. You can't say this is the best vaccine at this stage, right? You can't say this is a global vaccine that will cover all populations. Remember, in infectious diseases, it's a function of 3 things. It's the organism, the host, and the intervention. So this organism is a moving target. It keeps on mutating. It keeps on resisting what you're throwing at it. So you will need to look at all those factors when you are choosing your vaccine, and there's no one better than the other. But you will need a surveillance a program there. And then the other consideration, which is very important for our health systems, right, is does this vaccine protect against severe disease? Does it protect against hospitalization? Because once it does not protect against hospitalization, I don't think it will be a very useful vaccine, because you then congest the hospitals with COVID, and then the other programs suffer. People with diabetes have no access to treatment. People with cardiovascular conditions have no access to treatment. Day surgeries here have been closed for the past, they only opened recently, last week or the other week. But since December, no day surgery or elective surgery was being performed. So those are quite key considerations when you are going to choose your vaccine. And at this stage, I won't say there is any better than the other vaccine. Ejitio will tell us about the UK experience and what they have done with these vaccines, because they've got an active program that's running there now.
I'll come to that in my lecture, Becky. Thank you very much. That was a fantastic lecture. The question I had for you is based on the governmental extensions of duration between first and second vaccination. What is your view, considering the studies that have been done and then governmental decisions to lengthen durations?
I was hoping you wouldn't ask that question. Right. I think in one of the clinical trials with, I think it's with the I think it's the AstraZeneca vaccine, where you got a more profound immune response if you delayed the second vaccination by up to 12 weeks. Right. So you actually have a benefit from a physiological benefit by delaying that. Then there is also a logistical a benefit in that you vaccinate a larger population. And what are the protections from the first dose? Now, the protections from the first dose vary from what vaccine you are using, right? Novavax vaccine and the and I can remember this vaccine, you get about 40 to 60% protection with the initial dose. So in a shipwreck situation where you've got a canoe with one oar, you cannot say, I can't use this canoe, I will rather have two oars before I use a canoe. So it's a delicate balance. There is scientific evidence to support the prolongation of the second dose. And there is a very big logistical reason. The factories cannot cope with the manufacture at this stage in the volumes that are wanted. And particularly now that you've got these variants emerging, right, you will start needing to alter the formulations of the vaccines. There is no perfect answer to that question, unfortunately, HD.
Okay. Thank you, Dr. Magazi. Thank you very much for your presentation. I think for the sake of time, we'll move on. I hope we have some time later on for more questions. And also, Professor Kerina Dori is going to speak. She's also coming from a scientific background. I'm sure she'll be able to answer some of the questions as well. So thank you very much, Becky. I think we'll allow Professor Dori to share a presentation, and then we carry on. Professor Dori is with the University of Zimbabwe. Her basic training was at UZ, but she went on to specialize in immunology and virology in Norway. So, Professor Dori, if you are there, please can you share your presentation and start?
Good morning.
Morning. Hello, how are you?
Thank you. Thank you for the invitation. Okay, thank you for the invitation. Mine is more of I was tasked to talk about how these vaccines work, more from a basic science perspective. I don't have much real clinical hands on COVID-19. My background, I'm working more on HIV. So mine is sort of really basic science talk. Thank you.
Thank you very much.
Yeah. So it's sort of a refresher course. Yeah, so this is my slide. Okay, so I wish to okay, so I just want to sort of to simplify this, the vaccine jargon, and also to understand how the 6 classes of more than 60 COVID vaccines work according to Dr. Mwale's excellent presentation. So I'll concentrate more on the 6 which are already on the market, how they work. So that probably, since in Zimbabwe, I think vaccination is voluntary, I don't know which ones will be brought in by the government, but I know of the Sino vaccine which will be coming, but for people to be informed of this different antigen preparation of these 6 classes of vaccines. So what I'm going to do, I want to talk a little bit about the background, which has already been covered by Dr. Mwale. I'll probably rush on those slides. And I think history is also quite important, because I want to also walk you through the early, what has been happening since Dr. Jenner's preparation and also how it has been revolutionized to the genetic vaccines now. I hope you are seeing my slides.
Yeah, if you could put on slideshow, please, Prof.
Okay, sorry about that. Okay, so and a little bit about the adjuvants. I think also Dr. Mwale. So it's also sort of overlapping, but Dr. Mwale was more clinical, and also some of the sticking issues, which I think is they are critical about also correlates of protection, stability, and also most of the things also issues have been also been discussed by the previous speaker. So probably I thought, okay, this is really basic about active immunization and artificial, which you want to talk more. Oh, sorry. I don't know what is happening to my slide. Sorry. Okay, the artificial one and the natural one, the natural immunization, which we get through natural infection, which I think is also a subject of interest, more so with this community transmission which we have. And this may also have a bearing on how our vaccines are going to perform in the artificial one, and also about the passive immunization of COVID virus a little bit, but not much on that one. Sorry, my technology is failing me a bit here. Okay. So like what I'm saying, more is the aim of vaccine development is sort of to alter the pathogen, like the inactivated or the weakened one, or toxins if we are talking about the vaccines from bacteria, so that it becomes harmless, so that it's able to mount an immune response to B cells, to T cells, so that we are protected, and in the event we eradicate diseases. So what is also important is also about specificity and memory. So if you can also see, I don't know how to, you can see my cursor, that also during the initial immune response, the primary immune response, we have this lag phase, which is very prolonged, which is sort of reduced during a subsequent a response to vaccines. When we get a subsequent encounter with antigen during actual infection, or then we have the lag phase also. So like also what Dr. Male said, we also talk about the innate immunity, which is genetic. We cannot do much about it, but it's also very important that we understand the innate immunity aspect, which not many people are looking at. Most of the vaccines, we are now talking more about this in the innate immunity aspect, which is in pink on my slides, about the natural killer cells and the complement, how it also affects this COVID. Not much work is being done. It's a lot of work now on the adaptive immune system, the B cells and the T cells, which I think is really commendable. It's a lot of work going on around that. But I think we should also look at the innate immunity aspect. So I've also talked about the coronavirus phylogenetics, but what I want to emphasize, which was not discussed, is the unique aspect of this coronavirus too, that the particles are shared during the pre-asymptomatic phase of infection as compared to the other human coronavirus, which makes it a bit difficult to control. And that's why we have this problem we have. Okay, the other thing is not, we've already talked, doctor had a slide on Zimbabwe cases. So, but what is important now, the vaccines are important because of this huge economic damage, and people are getting poorer. more so in Zimbabwe, when people are self-employed, they have to move about, and also the social cost, and also the mental health issue aspect, which is also kicking in, is also important. So this issue of vaccinology to development is important, and the impressive speed which we've observed is very good. It has never been seen before, the speed of development of this vaccine. So I will not also dwell much about the spike protein. We've also, but I want to also emphasize the spike protein and how it is used to enter the cell, and what we have, the angiotensin converting enzyme on the host cell. We are going to, and also what is important here, since some of the vaccines use the subunit 1, the receptor binding domain, which also interact with this, is also important to facilitate viral attachment. So from the historical perspective, we also talk about Jenner, Edward Jenner, about the smallpox. This is the first person who also developed in the 18th century, and how he used different vaccine, which we are already seeing even in the heterologous platform. That is 2 vaccine so we are using 2 vaccines to also come out with an effective vaccine. So in this case, he also used sort of a milder vaccine to raise an immune response to protect against the more virulent vaccine. So it's not something very new. Some of the preparations of using 2 vaccines, 2 viruses to come out with an effective vaccines. So that's the other thing. So using Dr. Jenner's finding, then we also talked about Pasteur, the French microbiologist, who also observed that this is the issue about attenuation, which you are also talking about, the sign of vaccine, which you are talking about. But of course, it's the processes of attenuation probably which are different. So in this case, during the 18th century, they just observed that time. They just observed that the old cultures sort of lost their virulence, probably due to mutations, such that when they try to make this chicken cholera vaccine, the chickens no longer died from the virulent strain. So the issue about attenuation, the issue about weakening the vaccine is not nothing, it's not anything new. So it has been done before. Probably it's the technology that has improved over the years. So by then the attenuation, so the viruses, like what I said, they are parasites, like the previous speaker. So they replicate so quickly, and the mutation rate is very high because of the polymerase enzyme, which it used with very reduced proofreading activities. So because we are also making this is what is also causing some of this mutation. So it's the mutation, and also passing the virus through different species is also been part of a strategy to weaken or to decrease the virulence of the viruses, sorry. So, and also can also use the recombinant DNA strategies, in which you can also isolate and also delete the virulence gene or mutate it so that it's now harmless. So you can also do more things in the lab, which were done, which took time, but when you do it in the lab, it also is faster. And like when you can also alter all these temperatures and also anything. And the good example also about changing the environment is the BCG vaccine, in which the addition of bile in the media also affected the virulence. So there's also a lot of science also to attenuate or weaken these viruses, which I think can be done. So I think these are sort of the different antigen preparation for almost all the virus which we have, including the COVID-19, the natural. Okay, you can have some immunity. Some of the viruses, once infected, like the measles, you are infected, you are protected for life. So for the natural infection with the COVID, that's, I think, the other sticking point. We don't really know. Of course, we know you are protected for at least about 5 to 6 months. That's what the literature is saying. So, but so we don't really know how of course the half-lives is not been sort of being researched around in different environment, because like what the host, the environment also plays a role in the comorbidities and also all these other things. So we have also a lot of issues around natural infection, more so around asymptomatic COVID infection. I think we should also look into that. So the attenuated, I think this is, we have some vaccine, which we have an example of COVID, and also the dead live, the dead, of course, I don't think we have a candidate there for SARS, then but the subunit down there in the recombinant, most of our preparations are recombinant DNA-based antigen production for the SARS, for the COVID-19 vaccines. So once you have this, your antigen preparation done, so there's also this issue about scaling up this antigen production in the purification, which previously we used to have production in animal tissues, which we now know it has been really a challenge to do with expense, keeping the animals, and the issues to do with the zoonosis. And I think a big example is this the polio vaccine, which was grown in monkey kidneys and was contaminated by the SV40, this monkey virus. And I think that's why we've moved from this animal tissue to human cell lines. Because if you also look on misinformation on media, you also see a lot of people also talking why we are also using these human cell lines. So most of also our vaccines, including the Sino, the Oxford, the Seneca, they also use these cell lines from the HEC and also the MRC and so on, which are also from aborted fetuses some years back. But this also raises a lot of some ethical issues with some, probably the Catholics and some anti-abortive movements. But what is important about these cell lines, they sort of have, there'll be small factories to sort of to produce these quantities of virus antigens in big quantities. So, and also then you have also to purify them so that we don't have any contaminants of human tissue in the vaccines. So it's a lot of, and it's quite expensive. But the more we purify it, we also run to the issue of losing immunogenicity of these antigens. So that's when the issue of adjuvant, which was also mentioned by Dr. Magazi, kick in. So the adjuvant, so we want to increase antibody production. So there are different type of adjuvants, depending, some just oil in water, or you can put some mycobacterium some mycobacterium constituents there, and aluminium. I think that's the main one, the aluminium one, the alum, and also some can also have some immune stimulatory complexes, sort of to boost the immune response. So the types of COVID vaccines, basically I classify them into 6. The messenger RNA vaccines, non-amplifying. Then you can also have the self-amplifying messenger RNA vaccines. But now there's none of this, the self-amplifying one is now on the market, but I think you'll see why it's also important. Then you can also the viral vectors. Dr. Magwazi has talked much about those ones, and the protein subunit vectors. Most of our vaccines are protein subunit vaccines. I think a majority of our vaccines and potential candidates are around the protein subunit vaccines, and the inactivated whole virus vaccine, the Sino, and the DNA vaccines. We don't have anything much on these ones, but also for the RNA and the DNA, it's also important to remind you where transcription takes off or translation, when the DNA transcribes to DNA, or what happens, the protein the protein synthesis is done in the cytoplasm, so nothing in the nucleus, whereas there are so many myths about whether the messenger RNA can also be incorporated into the host genome. So then when we talk about the mRNA vaccines, Doctor has already talked about about it, so I will not take much of your time, but to highlight the aspect of lipid nanoparticles, which are also embedded on the messenger RNA to make it a little bit stable. And also, the lipids also act as adjuvants. So then, like what I'm saying, is the translation aspect. We don't need to care about the nucleus in this aspect, about this integration of viral genome into the host genome. And this is quite faster and cheaper. It can be done into the lab, and it can also be easily modified, the mutate, to accommodate these mutant strains, as previously highlighted by the previous speaker. So I think this is what I'm trying to say. So if we have your gene sequence, whatever variants, the UK variants and other variants to come, we just package them in these nanoparticles and then express them without taking much time. So I think it would be much easier to cater for the mutants, strains of the COVID-19 virus. So these are the examples, like what he said, the previous speaker, the Moderna virus and the Pfizer are based on the messenger RNA virus, but of course there's some mild adverse reactions, and also which may occur following the booster doses. So I thought it was also nice to give the examples for, then there are also others, there are so many in candidates, messenger RNA-based COVID-19 candidates still being researched on all over the world. But you can also see, I don't know whether it is this other, some companies that has also produced a MERS virus based on the messenger RNA, also still working on the COVID, the Japanese one on this slide. So then we also go to the other classes, the viral vector vaccines. It's also, most of them, they are based on the viral vector, mainly the adenovirus. So it's also not new. The viral vectors started as early as the early 1970s, and we already have some vaccines already using this preparation, the Zika, the flu, the Ebola virus. So nothing is really new other than the messenger RNA. So you have the spike protein. So it has been made harmless by deletion of this early region, which makes it able to replicate. So it's quite safe. And you can also have the other more effective adenovirus, different strains, to make it more harmless and safer to deliver the immunogenic antigen. So we can also have examples of vaccines using the viral vector. It's already saying, we've already talked about it. And the issue to overcome the pre-existing immunity against this adenovirus following upper respiratory infection have already been discussed. And that's why you also have this heterologous boost strategy in which you can also use a different adenovirus from our cousins, the gorillas, the chimpanzees. Then you can also see that the AstraZeneca virus vaccine used the chimpanzee boost also to circumvent this challenge of pre-existing immunity. So the advantages also, these viral vectored vaccines, they can be, the expression of protein is quite high and the induction of the immune response is quite strong. And also, like what I said, it's not, they do not affect or interact with the host DNA, which is a also of misinformation which is circulating. So the inactivated vaccine, so it's also the Sino. So they've inactivated that virus, but it's also nice because, of course, if it's inactivated, we are not concentrated on the spike protein also. We can also get an immune response to other antigens that which is not really been so far been researched. So I think it also leaves room for us to can get other immune response. So it's nice as we get this sinovirus also to do some research to see the titers of antibodies and the specific antibodies produced after administering this whole inactivated virus. But as the previous speaker said, it's not appropriate for people who are immunocompromised. So these are also more examples of inactivated COVID candidates. It's a lot of them in preclinical stages. But okay, but also for the KM biologics, they also have the inactivated virus. We have the Zika virus, which is also already on the market. Then the attenuated virus is also some more examples. Then the protein subunit, okay, so it's also another example, which is the synthetic subunit approach lies in between the nucleic acid and the inactivated virus vaccines. So it also recombinant and also typically also expressed in cell lines, including the insect cell lines. So we have this Novavax vaccines as an example for that platform. So it's a lot of all this still in preclinical development, a lot of candidates. Then you can also have other classes which are not yet on the market, the DNA viruses, and also more interestingly, the virus-like particles in which we are using informatics or bioinformatics analysis to map sort of the immunogenic, the epitopes and the immunogenic response so that you can also produce precise the antigen that are more immunogenic. So it's also a new, so we have also a lot of candidates, a vaccine using this. Okay, so this is an example of the DNA platforms, already candidates using the DNA and the then the concern about the DNA vaccine, which says there are some concerns that it may be integrated in the host chromosome and may turn on and off some tumor oncogene or tumor suppressor genes. Then these are examples of the virus-like particles. It's a lot of also some virus candidates also using this platform. Then also, I thought it's also nice to talk a little bit about this, some sticking issues in this COVID vaccine development, the issues to make the current messenger RNA virus more stable, more so here in the developed country. I think it's a pie in the sky to have them if they need these -70 temperatures. So I think we also have a lot of research around additives that may also being used to extend the vaccine shelf life, and also to have it lyophilized or freeze dry them so that you can just reconstitute a powder before injection. So we have a California-based company also working on this strategy, and also to make it the shelf life easier. I mean, that's the logistics. Then the issue about we talked about the messenger RNA, but there's also the issue about the self-amplifying messenger RNA, in which the self-amplifying, like what he's saying, it's also used in this plasmid, the Venezuelan equipped encephalitis virus. So what is it? Why is it important? It's also important because we may also run away from all these second doses or third doses because it will be self-amplifying. And also there's also the issue to reduce the amount of vaccine or the volume of vaccine that can be administered into the arm. So far, like the messenger RNA doses range from 30 micrograms for the Pfizer vaccine to 100 micrograms to the Moderna virus. So it's a lot of, the mass is quite huge. Then you can have this CureVac, which is 30 microgram. So what we are saying about this self-amplifying messenger RNA can also overcome these issues to do with the high volume of vaccine doses. So the vector is engineered so that it can also make more copies and it self-replicates. So that's the other issues. Then the issue about circulating variants is also an issue, like what I said, the viruses are very unstable, mutating. So the previous speaker has already talked about this variants, so I will not take much of your time on this one, in this slide. But also in Zimbabwe, I also understand we also have some surveillance system which also being put in place at our national reference lab in collaboration with our UK partners, which I think is also very important. Is these mutants also like the potential consequences is also being discussed. So I feel I'm also repeating ability, transmissibility, severity of diseases, and also these issues to do with false negatives, which I think is really an issue so far, even in Zimbabwe. I mean, most of even my friends and my families with clinical disease, but they have false positive PCR results. And also, we can also have this issue about evasion of natural or induced immunity because of this immunity, of this mutation. So this is also quite a concern, given that we are vaccinating a large proportion of the population globally. I think it will be also an issue in the long term. And also, how long, okay, the other issue is also, some people are also very skeptical about how fast the COVID vaccine has been produced compared to the traditional other vaccines, which is an impressive 1 year compared to the polio vaccine that took almost 50 years, and the measles 30 years, and so on. And also some of the infection like HIV is 40 years now, and we are still not, we are still going, we don't have any vaccines. So I think I also get some misinformation about how also people think, why is it so fast we got this COVID vaccine in 1 year? But I think it's also due to that the COVID vaccine development, because it's a global problem, it was well funded and it was really produced and very fast. And also, there are also some issues about the use of human cell lines, the fetal one, more so to do with the MRSA, sort of the aborted fetus cell lines. So the Catholics and some people may not also be willing to take it up. So I thought it's also nice to say, I mean, depending on your belief and so on, these are some of the things which some people may also want to consider before they choose their vaccine of interest, because since here also in Zimbabwe, it's also voluntary, and I hope we can be obvious, we will have the opportunity to choose if we have the option. So this is what these are the features of an effective vaccine. So what is now important is if we can also have a tick box. So about safety, safety and protectiveness. I think this, since they passed the phase 3 trial, the phase 3 clinical trials, the issue about safety and protectiveness, it's a tick and a tick there. But the issues to do with sustained protection. So I think this is the challenge which we have currently with our vaccines. Of course, we know they are effective 95%, whatever, but the longevity, how long we will be protected, we still don't know. And the correlates of protection, we don't know. And so the issues to do with the antibodies and the T cells, I think also the previous speaker touched on that. And also the practical aspect about the cost, the biological stability, ease of administration, the storage and transportation, and the side effects are some. What I heard someone who was asking about which criteria which we can use to select the vaccine which may be suited for our situation. So I think this slide may also partly answer that question. So, and also this issue to do with herd immunity. I think it's also important that I think it's also not necessary to vaccinate 100%, because if we reach about 70%, we have reduced the transmission rates. The virus is less efficient to transmit, and eventually the infection will not maintain itself. And this is what is sort of what we mean when we talk about herd immunity. So I think which is a good news. So since we don't have to vaccinate everyone, so who is to be vaccinated and why and how, I think is also the public health aspect, I think, but it's obvious, targeting the people who are at risk and so on and so on. So I think this is the end of my presentation. Thank you.
Thank you very much, Professor Duri. Thanks for teaching us the science of these vaccines. I think it's very important that we understand the basics about the vaccines for us to make good decisions. So let's now move on to questions. If there are any questions for Prof, please go ahead. Yes, Mr. Macheka, you can go ahead.
Thank you very much, Prof, for that very interesting presentation, which takes some of us slow learners through the basics of this vaccination process. Your very last slide touched on herd immunity, and the previous one was talking about the Sinovac, and that is an inactivated virus, and that is deemed unsuitable for the immunocompromised. The problem with that is sometimes it's taken as a very loose term. When you say immunocompromised, is there a level of immunocompromise where you say this one, for example, with your experience with your HIV cases, who would you say can receive it and who would you say cannot receive it? Or is it that all those who may potentially be immunocompromised must not be exposed to that kind of virus and rely on herd immunity? Thank you.
Thank you for the question. Yeah, I think immunocompromised, I think we can classify for HIV, I think we'll be talking of those people with AIDS. I mean, with the CD4 count probably less than 300 or so. I'm not sure whether there have been some studies which you can also attest to that. But if you are really sick, and I think we should have sort of a cutoff CD4 when, but now I don't know for HIV, I think we have sort of a cutoff for the CD4 count if it's less than 350, I think 300 to 350, or some patients on 250, I think, or some patients on cytotoxic drugs, or the cancer patients, probably they should also be discouraged probably to take those the vaccines. Yeah, I think probably that's what I can also say. But if you are immunocompromised, but you're still immune competent, yeah, because it's about if your immune system is still not that down, I think you can still take the virus. Just like even our babies, the infected babies, HIV infected babies, they still take these IPI vaccines, and I think they are still doing very well.
Okay, thank you very much.
I'm not sure whether I answered your question.
Yes, I think it did clarify some issues for me. Thank you very much.
Hello, so are there any other questions?
Ichikari, just to add on what Prof has just said then, the issue is when we say with live attenuated vaccines, you should use them with caution in the immunocompromised. The thresholds for immunocompromise are not exactly well defined, but the concern there is one, you might not get the immune response or the neutralization antibodies that you desire because of the immunocompromised state. And two, you might get vaccine-induced disease. And with the current attenuation processes, the second concern does not falls away, but not entirely. So that's why we say use with caution. We've got this experience from yellow fever vaccination, particularly here in Africa, where we've got high HIV burdens. We still vaccinate people against yellow fever without much of and achieving good antibody titers and without activation of yellow fever disease. So it's a cautionary statement that you use with caution in the immunocompromised for those reasons.
Sure, thank you for that, Becky. Thank you for the clarification. Okay. Right, so thank you very much, Prof. I think we'll move on for the sake of time. Let's move on and with our next presenter. Egidio da Silva is a consultant anesthetist in the United Kingdom. He is the son of the soil. He trained here initially at the medical school, and then went on to the UK to train in anesthesia and perioperative medicine. He was involved recently in a program to administer vaccines to, I think, some 5,000 health workers in the UK. So he's got a lot of experience with clinical handling of the vaccine issues. So Egidio, if you could please kindly share your screen and then make your presentation.
Hello, I am just going to go to. Is the screen visible, guys? Sorry.
Yes, we can see your screen very well.
Okay, thank you. So I'm coming at this from a perspective of really process development, process mapping. And my experience is more from the IT support to vaccination hubs point of view. The total, sorry, I'll just go to the previous screen. I do have to do some declarations and just skip to that. So coronavirus in the UK, this was from the 10th of February. The total deaths by that point, so this is a couple of days ago, 114,000, just under 115,000. The total cases that we'd shown were just under 4 million. Vaccination doses, the first vaccine given was just over 13 million. And people in hospital were 26,000. Now, just 12 hours later, the deaths had increased. We had a few people in hospital, fewer than 12 hours before. The total number of cases had increased slightly. Vaccinations, understandably, had increased slightly. So things you could argue are going in the right direction. Now, I work in Birmingham, and from the Birmingham point of view, we've got the perspective of what is showing up as the national figures, the national status. And then we have the local status. And the local status is the University Hospital Birmingham's are divided into 4 main hospitals, and each of those will show up as total positives, total tested, current positives in patient. The figures, as you can see, are quite high over the period. The total deaths since this all started, just in our group of hospitals, is 2,409, and that was the day before yesterday. That's increased slightly now. The geographical distribution, I've put a big black arrow there of where I live in the middle of all of this, which, yes, it has been scary and it's been challenging. So just down to the hospital that I'm doing a little bit of ITU in, there's a racial distribution, which is not exactly completely reflective of the population distribution. And that's why we know that our BAMI group of patients at higher risk. As you can see, the number of Pakistanis are much higher in relation to our admission rate than either Afro-Caribbeans or Indians. The other group are, some of them may actually fit into these other groups, but just don't define their racial heritage on admission. I know Becky has spoken about this, but I'm just going to go to the point of the vaccinations and the cost. So when this all started in the UK, the first one to be approved was the Pfizer-BioNTech, and that was in December, followed by the AstraZeneca, which is the Oxford version, followed by Moderna. And the costs are listed there. The other one they're looking at or have been looking at recently is the Sputnik, which is half the price of the Pfizer and has a better fridge storage. which is something I'll come back to about fridge storage and duration and temperature. And I think Becky already talked about it, but it's worth just revisiting. So this is a website that was launched on the 13th of March 2020. The Perioperative Medicine, the Royal College of Anaesthetists, took ownership of this, and I've been getting emails and requests from colleagues in Zimbabwe and around about policies and process. And I think this is one that when you click on that point, this is the page it takes you to. So I'm just going to stop at this page for a short while and take you to it. And I hope you can see the screen share that's happening now. So it takes you to this page. There's the background. If anyone is looking for development of clinical guidance, the clinical guidance then takes you to all of these different ports, from airway management to obstetric. And whilst it says, for example, obstetric anesthesia, it does give you just obstetric advice on infection prevention, infection control, drug supply and demand. And an important thing that we found is well-being of the doctor, of the healthcare worker during COVID-19. And that's important. I'm asking the question if you're okay yourself. And I mean, personally, we've had WhatsApp groups set up, whether it's with my original class of qualification or the people I work with. It does help with offloading of the psychological stresses of the day. Protective personal equipment and shielding and understanding those elements. Pediatric management, again, is important. Restarting planned surgery is something that our patients keep asking, and that, I'll just click on that one, for example, and it'll take you to advice and policies on each of those, of how to, for example, aerosol clearance times, and how you can effectively operate with better aerosol clearance in aerosol generating procedures. Cross-skilling, so we're having surgeons or people that are not actually having current clinics working in teams in intensive care, supporting either medical staff or nursing staff. So these are elements of where we can find the clinical elements. There's national guidance here as well. So clicking on the national guidance, it takes you to a whole list of things of how to deal with pregnant employees, how to have strategy with coronavirus in the workplace, the coroner's guidance, the laws that come up with that. You can have patient information, and again, that gives you similar advice. So these are quite useful tools. I'll just take it back to my screen. The other thing that's really helped, and I think I'm not sure how much has changed in Zimbabwe with regard to a coronavirus act, but this, when you look into it and sort of look at the wording, it does help. It forgives a multitude of sins, but it does protect the healthcare worker without covering up anything sinister. So these are things that prevent or reduce the amount of future potential litigation. And these are things that are worth looking into. Now, from the IT point of view, we've had primary care teams admit and govern the care, the people that actually decide whether a patient, when they start on dexamethasone or when they start on tocilizumab, when they need to be prone for ventilation. We then had procedure teams who are doing lines, tracheostomies, intubation. We have proning teams. The first wave, there was complete pandemonium. As we started organizing these teams, we learned how to do this better as a hospital, as a country. Now it's better organized. The proning teams come in at certain times, the procedure teams come in at certain times. You have teams that transfer, so when they're not proning, they'll probably transfer a patient to a CT or to theatre for a tracheostomy if it's complex. If it's not, it's done on the unit. But again, you want to reduce aerosol generation, so you take it to a laminar flow setting. Then you have repatriation teams where you transfer patients to hospitals which have fewer patients and therefore more potential for looking after critically ill patients. The vaccination program. So this is something we started setting up, and I know Becky and Prof. Jury have already touched on this. The cold chain general principles. I had a document that was written, sent to me by a colleague, Dr. Mgodi, which was written by Dr. Hove, and that actually covers all of this point. But I was quite impressed by the fact that all of that is projected to cost under $7 million, whereas ours are in the hundreds of millions when they're projected here. So the storage and transport becomes important, and the type of vaccine you choose. Systems and equipment, again, for example, with the Pfizer being stored at -70 degrees, being transported in lorries that can afford that sort of negative temperature. Actions to be taken if incorrect storage or transport and how that's dealt with. How much the portable refrigeration costs, who owns it, how it's procured, the staff that you have. So if people are not working in theatres or in a clinic setting because of lockdowns or because of reduced functioning, those people can then be afforded the opportunity to work in vaccination programs. We have had in our setting a couple of commencements of litigation where you have patients who feel they've been injected a bit too high on the shoulder, allege that they have reduced ranges of motion, frozen shoulders, nerve injuries, neuropathies, and that's something that's unavoidable and we just need to cover for. It's a more litigious society in the UK and in the States, so this will happen. Now I've put up a card there, and this is very valuable currency at the moment. These are things that are that can be misappropriated, that can be stolen, that can be sold for more money on the black market, because people can obviously will need these sorts of things as proof that they have been vaccinated to travel. Well, that's the projection. And as such, it is a good idea to sort of, in a way, control these documents. The other thing that we found is, thankfully, we haven't had it at our centre, but we've had it at other centres where used vials, have been stolen and are being used by con artists to suggest that they're providing early vaccinations to patients, elderly patients, by visiting them at home. A link that I've put down there is for advice and guidance of what advice and what information we give to patients. It's available in the Green Book, Chapter 14A. The link is there, which takes you to the Green Book, and then just waiting for that to come up. Sorry. And that then enables you, or just makes it easier in these busy times, to use a lot of this documentation with reference studies on how decisions are made and how what information we give to patients, and the amount of information there is quite prominent. It's just about what we choose from it. So with the vaccinations, we are keeping cumulative total, which is available on the website nationally. The second cumulative total, so this one we're at, well, now it's over 14 million. The second doses, we've had just over 500,000 patients that have already received the second dose. I have put the common side effects. Now, I had the Pfizer vaccine. Personally, I didn't really have any of the side effects except a cracking headache, which is indescribable. It lasts for a few seconds and goes away. It's almost like a spasmodic headache. It lasted, for me, it lasted for 24 hours and then it just went away. Paracetamol helped quite a lot. What we have a lot of people reporting is the tiredness element of it, headaches, muscle pains, joint pains, and not so much fever, but it does happen. The reason I've put there beware social media is we have the current rise of the social influencers, and these influencers describe side effects and sometimes embellish them to a point that it's made to sound really horrible with affiliated pictures to go with it. And as a result of these things on Twitter, Facebook, Instagram, you name it, the uptake of the vaccine then drops, and that causes, it's an element of scaremongering. The AstraZeneca, this is the one that we had experience with giving a lot of. I didn't have this, but colleagues that had it, the biggest thing that came out was people, for the first 24 hours, there is a feeling of feverishness and chills. Headache is not as common as with the Pfizer. Feeling tired. So a lot of out of our hospital health workers, at least 50% described just fatigue. And that fatigue sort of has an onset from the evening of the injection to the next day. And then it goes away as quickly as it's come. Usage of paracetamol before and after the vaccination has helped and alleviated those symptoms. And people that have done that religiously have avoided having a huge amount of side effects. We've got global range of vaccine skepticism, and the thing with that is it's going to happen. It's going to happen in Zimbabwe as well. There are people that watch social media, the people that listen to conspiracy theories, and they will say no to the vaccine. And I'm sure the fact of herd immunity and the fact that between 70 and 75% of population should be vaccinated to try and improve or influence the chances of herd immunity are necessary. But when you have patients turning it down on grand scales, it becomes a bit worrying. The only thing that sharpens the focus on having the vaccinations, people understanding that there's the potential that they may not be allowed to travel, that they may not be allowed to enter certain big buildings unless they're carrying passports. And whilst that's being talked about, it's a potential. So our local experience, 5,000, just over 5,000 vaccinations in our staff, 50% reported side effects. We had to have an on-site medic, and I think this was because it was an unknown quantity at the time. So initially we used to have a consultant physician or consultant anaesthetist. Now we have a registrar level doctor around. We have 8 lanes, they're 15 minutes apart to allow for social distancing. Out of that 5,000 patients, we didn't have any anaphylactic cases to date. The patients were advised to wait in the waiting area for 15 minutes initially when they started However, government guidance has come out that for AstraZeneca, we don't expect that. However, for Pfizer, that continues to happen. I might have said 3 patients had cloferinumab, that's for expected, just very slight rashes, but more as a preventative measure. We've had 2 points of patients starting litigation about the cause of frozen shoulder because the injection was too high. And in our setting, that's expected. So I've put that slide back up because it's quite interesting that we have a £3 vaccine, a £15 vaccine. Understandably, that was approved first, so we're paying for that. But what's interesting is our government, even if we have 2 vaccines necessary per person, we've ordered 357 million vaccines. Now, this is a government. This is on the government website. We've ordered all of these vaccines. We've invested over £350 million. Now, a population at most around 80, even if we gave 2 vaccines per person, that's 160 million. So that begs a few questions. Additional doses have been ordered. That's the cost of what we're spending. There have been links, and this is going to be in every country where people can make money. It's just human nature, where links have been shown, in the financial aspect. So what is our medical responsibility, irrespective of how people benefit from things like that or not, is to understand the potential for improving herd immunities, to allay fears, provide the correct information, encourage simple behavioral modification, enable and set up vaccination programs. And those that are not doing clinic can be harnessed for use and help in these programs. So the questions are, how many vaccinations do you actually need? What is the ordering administration system? Are transport and refrigeration systems in place? Some of those questions were answered by a document that was sent to me. Certification systems, it is an important currency and it will cost. Schools as vaccination centers, that's a possibility depending on whether the kids are going to school or not. So we outline our pathways which are being done. Nail down procurement because there is corrupt procurement everywhere or misplaced procurement. Are medical people given the opportunity to be part of this, or are they marginalized by government? The nature of entrepreneurship innovation, does it end? I put that down from memories from medical school, whether it ends at Warren Park or whether it's actually something that happens and we're listened to. And then, the potential differences. And I hope that the links that I've put in have helped. That is pretty much what I had to say, but I'm open to questions. Thank you very much.
Thanks, EJ. Thanks for the very wonderful presentation. Thanks also for sharing your experiences in the UK. I think it will help us a lot to know that you guys have gone through this. And it looks like it's not as scary as we thought. If there are any questions, please go ahead and ask.
Yeah, hello. I think I have a question here from Mr. Speaker. Thank you for thank you for the presentation. My question is on mixing vaccines. Let's say you have received, it's now with the South African variant, which is more like infectious, and we know that the AstraZeneca is not very effective. Let's say you have received the first dose. What are your thoughts on mixing the vaccine so that you don't get, let's say you don't want the AstraZeneca one and you want a different one. What are your thoughts on that?
I think that's a question I would rather defer to either Becky or Prof. Judy, because from my point of view, it's something that's being examined, it's something that's being looked into. I haven't seen any conclusive studies yet, but if Becky wants to answer that, if he's still on the call.
Thanks, EJ. You actually asked that question a couple of weeks ago. There are actually clinical trials that have started now looking at the impact of mixing vaccines. But we should also be aware that with the Sputnik, with the Russian vaccine, it's basically two different vaccines, right? Because you are looking at the adenovirus 26 on your first choice, on your first dose, and you are looking at adenovirus 5 on your second dose. And that's what we call heterologous inoculation. Now, the idea there was to bypass the impact of neutralizing antibodies against the vector, right? So that you can actually have an immune response to the antigen and boost it with the second dose. So with that concept in mind, that's what is informing the current thinking in the clinical trials to see, can we get the same effect if you used a protein-based and an mRNA-based virus at this in two different inoculations? But currently there is no, the jury is still out. The clinical trials have started in a couple of centers to look at that question.
If I may add something, patients with autoimmune diseases are being excluded from these. And now some of you might have noticed that tocilizumab is been approved for use in intensive care. If I may just highlight what's happening with that, what we've noticed in intensive care use is the CRP, which is C-reactive protein, which is used as an infection marker. Drops within 24 hours. You have it from over 200 to less than 10 as a result of tocilizumab. So now what we're doing is, if we start that drug, we're using procalcitonin as the infection marker for bacteria, and galactomannan for viruses. Sorry, for fungus. Pardon my mistake there, for fungi. And for that, we're doing that every 2 weeks for the fungus, and the procalcitonin we're doing 3 times a week. That's if a patient is on tocilizumab. The antiviral we're using now of choice in a proven viral, sorry, let me get back. The antifungal we're using of choice is voriconazole. In proven fungal infections. Thanks.
Okay, okay, thank you. Are there any other questions? Mr. Mtewe wanted to ask a question. Okay, Ms. Macheka, please go ahead.
Thank you very much, Dr. Da Silva. You mentioned, in passing, one very important point, which is quite topical, and this is, the rich nations hoarding the the vaccine, and in particular, you indicated that the UK has nailed down 350 million doses for an 80 million population. Do you get the sense that this kind of procurement is being driven by, like, for example, the UK COVID task force, or it's a political issue? What sense do you get from this sort of action? What is the intention of that?
So personally, what I've done is I've challenged my local MP to find out this, and the response that's come back to me from government is that this is what has been ordered. This is not what is in the stores. And you can take this with a pinch of salt, because when politicians, it's something my father said to me a long time ago: politicians and nappies need to be changed often because they get dirty quickly. And this hasn't changed. Now, with regard to their argument or their explanation, is that they haven't actually procured this. This is what they've ordered, and depending on what they receive, that's what they're going to use. Now, I beg to differ, because if they've already set aside 230 million and spent it, I think they've procured a lot of this, or rather the order has been placed and is paid for. So the argument, the second argument they brought back is actually, you know, if there's stuff left over, it will be donated to other countries. Now, you know, again, another massive handful of salt with that one, but your guess is as good as mine.
Okay,
Thank you.
Tendai Suro, please ask your question.
Thank you very much for the opportunity. Thank you, Dr. De Silva, for that one. So my question basically is on whether there has been a proven or a tentatively proven difference in efficacy between, in fact, firstly, is it necessary to vaccinate individuals who've already been exposed to the virus, whether it is, and is there a difference if it is a mild, if they were mildly symptomatic, or if they were overtly symptomatic or hospitalized? Is there a regimen that is different for vaccinating individuals that are post-infection or already been exposed to the virus? Thank you.
Okay, so part of that question, I will, again, Becky, I'm sorry, I'll refer part of that question to you, but I'll answer the parts that I can. So from personal experience and with colleagues having received both types of vaccines, I have had colleagues who've received the Pfizer vaccine, who after 4 weeks have actually unfortunately been infected with COVID, but have not needed to be hospitalized now, have not actually been severe. The only reason they ended up being tested is that because they were in contact with somebody that was definitely positive and that was overtly positive, and they were exposed and then tested. They were asymptomatic, but they went through the process of self-isolation. Out of those colleagues, one of them had a previous infection before the vaccination. Now, from my point of view, guess is as good as mine as whether his own immunity or the vaccination helped him in being less, in demonstrating a less severe infection the second time round. So it doesn't prevent anybody from getting infected. It just seems to prevent the manifestation of that infection. But Becky, if you want to answer the other part of the question.
Yes, I'll go back to the basic knowledge on coronaviruses. From previous knowledge with the SARS coronavirus and with the MERS virus, and then with the other coronaviruses that cause mild disease like your respiratory flu-like symptoms, we know that the duration, your antibody response, the duration of protection rarely goes beyond 18 months. So it's not a durable kind of immune response. So yes, you will get reinfected, and yes, you will get disease. A more recent example is the immune escape that we see with the South African variant, for instance, the variant that's currently circulating in South Africa. That during the second wave, you saw patients that were infected in the first wave, which only happened in June, coming back in the second wave in November, October, November, December, infected with coronavirus, with high viral loads, and sometimes with even severe disease necessitating admission. So yes, you will need to vaccinate those people that have been infected before. And it's going to be looking.
So Becky, I'm going to push.
Looking into the future, we are going to be taking corona vaccinations in the same way as you do with the flu vaccine, as determined by what are the circulating variants and what is the impact of those circulating variants.
Okay, thank you very much.
Okay.
Listen, Dr. Mwale.
Hello, yes.
It's Dr. Mari.
Wanted to say something?
Yes.
Yes.
Thank you so much.
Go ahead, please. Go ahead. We'll take your question, last question.
Yeah, thank you so much. I just wanted to get a bit of a feel on the global impact of these variant viruses and the different vaccines for international travel. It sounds like we're going to get vaccines specific for the region where you're living, depending on the variant. But then how long is it going to take for us to open up the borders to travel? And some flights go through different regions, taking an example from UK to Dubai to South Africa and back. It might mean that we have to get 3 vaccines to cover you for all the variants.
Yeah. Hello, yes, anyone in the panel to try to answer that question? So that will be the last question for this. We need to move on.
Can I have a go and with what
go ahead.
With the knowledge that's coming from the variants, I think it's actually now going to it's the question that's been asked and that needs to be solved. Do we need a univalent or a multivalent vaccine? Meaning that you will need a vaccine that not only caters for the original strain, but also covers all known, current, and circulating mutations. And if you are looking at current vaccine development, that's where it's actually headed. We already, in terms of travel and requiring, I think a visa, somebody put it in here, you already have that kind of requirement for yellow fever. If you go to yellow fever prone countries, you actually need to get a yellow fever jab or yellow fever vaccination. But it's an evolving situation with COVID-19.
Okay, thank you very much. I think we'll move on. Our last presenter is going to be Dr. Mawomba. I hope she's been able to join us. Dr. Mawomba is the Chief Coordinator of COVID-19 response in the Cabinet and in the Office of the President as well. So essentially, effectively, she's in charge of our national response. So she's going to give us a talk, and then we have a chance to ask her questions if we wish. Dr. Mawomba, are you there?
Yes, I am. Good morning.
Morning. Thank you for coming on board.
You're more than welcome. Can you see my screen?
Yeah, I think it's coming up. It's coming up. Let's see.
I hope it will. Screen is paused. I wonder why. Apologies. These technologies can be something else. Let me just try again.
That's okay.
Where is it? Hey. Oops. Can you see it now?
Okay.
No.
Yeah, we can see your screen now.
Okay. Sorry about that. It's frozen on my side. Frozen on my side. Let me try again. Apologies. It won't be long anyway, so please just bear with me. Just bear with me.
That's all right.
Let me try again. Screen share. We want to share that one. Share that one. Okay, hope you can see it now.
Yes, we can see it now.
Perfect. Thank you. Thank you very much. So to our, there. Thank you. Thank you. Truly appreciate it. This is not going to be very long, really. And I truly appreciate you asking me initially. Of course, I had said I probably won't be able to prepare something. I put a few slides. This week has been really madness, working with Minister of Health and so on, trying to really get ready. You heard all the pronouncements and so on. So this particular slide is really just going to give you an appetite of what government is thinking and what we're doing, and then give you an opportunity to ask any questions. So first, really, we are very aware that the pandemic has caused untold suffering throughout the world. That's why we're here. And Zimbabwe has really not been spared. Last night, you saw the updates from Ministry of Health and Childcare with the huge numbers, cumulative numbers. But I want to draw your attention to the bottom one there, where the case fatality rate for Zimbabwe is 4.0% which is among the highest globally, not just regionally. A true concern indeed. So as we work to try and address this, we're looking at some of the major shifts in understanding the virus since the mid-2020, where we really had a spike coming down, going up and then coming down. We are very clear that the pre-symptomatic and asymptomatic persons transmit the virus. We've acknowledged that from you, the scientists. We have taken on board the antigen testing, which has improved our testing to be able to appreciate our epidemic and hence tailor our responses. Development of the variants that you have been talking a lot about, the UK, SA, Brazil, and others. And of course, the innovative ways of developing vaccines. Once again, you, the scientists, are talking about, and we're delighted that there are about 15 vaccines now in the late third clinical trials, going through the WHO evaluations, and so on and so on. Very important background for us to appreciate. We're also saying this second wave that is affecting us is so much bigger than our first one, and I'll show you the epic very briefly. And we feel very strongly, we know the super spreader events that we saw in December, there around, really made this horrible. And of course, the family gatherings as well. Recently, you heard the family which had 5 members just going. We think it's some of those gatherings that really contributed to that. So the need to prepare and prevent a potentially bigger third wave using 2 fronts is what we are looking at as government. The first prevention front is, of course, our current response strategy that includes the prevention, containment, mitigation strategies, structured around the 8 strategies or pillars, consistent with the WHO guidelines that you are all now very familiar with, looking at the surveillance, testing, case management, infection prevention, risk communication, procurement, logistics, ports of entry, and of course, this coordination that I'm also tasked to do. This quite clearly hasn't been enough, and hence we're looking now at the second prevention front, which is the deployment of vaccines. And that's the epic curve I just said I will show you just briefly to remind ourselves. The first wave we thought was really bad, but when we really got hit by the second wave here, quite clearly, we saw it. You don't even have to see this picture. You can just tell by colleagues who have passed on and so on, that this has really been bad. We're happy that things are going down now, but we're now reaching those dangerous levels again. Dangerous in the sense that when we got down with the first curve, people started relaxing. We're seeing that once again. While we're trying very hard to step up our initiatives, response, and so on and so on, we're also scratching our heads to see what else can we do. And of course, the introduction of the vaccine is one such area, knowing very well it's not a magic bullet. So to come back to what we are saying as government, you all heard the government direction on COVID-19 vaccines, the statement that was issued post-cabinet meeting on the 9th of February 2021. That was, the cabinet was on Tuesday. That was when it was announced. The intention is to reach 60% or thereabouts of the population. Choice of vaccine will be science-based with adequate research and findings guiding decision-making. Priority will be healthcare workers, the elderly, and other vulnerable groups, and it would be voluntary. I think that's very important to say. Vaccine to be sourced from multiple manufacturers through multiple financing arrangements, but with government taking the lead. So what is the National COVID-19 Vaccination Plan? This is the one that we've been working flat out on. Yesterday, meeting with the PMDs and culminating in the Deputy Minister's statement. And you also then saw some of the slides that Dr. Howe issued out there based on some of the meetings that we're having. The National Deployment and Vaccination Rollout Plan is a guiding document that provides a framework for deployment development, implementation, monitoring COVID-19 vaccines in the country, ensuring, let me emphasize, ensuring that both the plan and the related financing are well aligned to the Zimbabwe COVID-19 preparedness and response plans and other plans, and that implementation is fully integrated into the national governance mechanisms and implementation structures. And here we are thinking of the EPIs and so on. The plan covers 12 key strategic areas as outlined below for a successful rollout of the vaccine. And I summarize this here just to remind ourselves, Zimbabwe is working very hard to be science-driven, and science-driven does not mean we have to always do the researches or the science analysis. We are very much guided and depending on WHO, which brings in global scientists like some of you here to help guide nations to say, these are the areas that you really need to look at when you are actually rolling out, deploying of vaccines, and pay attention to this. So we've just taken that on board, and quite clearly, background, people usually don't think of that as a strategic strategy, but it actually is. Country context is so critical as we look and say, what is it that we need to do? And of course, you then have the regulatory processes, planning and coordination that we're doing this whole week, resource and funding. You can't just think you have it for free from somebody. The target populations that have been talked about, supply chain management, human resource, including the training, advocacy, communication, social mobilization. What we're doing here is actually part of that. And thank you so much for bringing me on to your platform. Vaccine safety, monitoring and management of adverse events following immunization, and of course, injection safety, very critical. Remember, we're saying we want to be science-driven. We want to make sure that we're also paying attention to these things that will inform how we move forward. Immunization monitoring systems, and of course, the surveillance itself is critical. And finally, we want to make sure that we're evaluating the introduction of the COVID-19 vaccine in order to really make sure we continue resetting our dial, if you like, continue strengthening, improving, and making sure we are indeed addressing what our population needs and not what we individuals or politicians or whoever thinks it's required. So really, I wanted to give that quick background. The next slides are really things that you've already seen, but I hope they will actually help us juggle our minds so that we are able to ask important and good questions that help us, you and I, to move forward. So the objective of our plan is really to enable high quality vaccination services and reduce morbidity and mortality due to COVID-19 disease, very much linking with the objective of our preparedness and response plan that has those 8 pillars I mentioned about, which really focuses on, compacting, or what's the word I want to use, looking at the impacts, the adverse impacts of COVID-19, but with a focus on morbidity and mortality. Quite clearly, we know that if we don't do that, we will not have an economy to talk about. We need to first of all focus on morbidity and mortality. And hence, if a vaccine is really trying to do that, I think you are the scientists, you can tell me that as well. And of course, some of the specific objectives, which have a bit of administrative issues as well, is really to vaccinate eligible population on a voluntary basis, for free, vaccinate a minimum of 60%, as cabinet indicated, of the total population to achieve herd population immunity. And by the way, if you're taking away the 15 years and below, you can almost say we are literally going to vaccinate everybody. But as you are aware, as long as we're talking it to be voluntary, there quite clearly will be some people who will not want to be vaccinated. And hence, we are sincerely hoping we will get the percentage that we want to be able to get that population immunity. And of course, we want to initiate vaccination through eligible high-risk target populations. I've already mentioned it. Adequate vaccine supplies for the activity. Some of those are really more administrative issues, if you like. So the regulatory framework, the COVID-19 vaccine to be registered by Medicines Control Authority of Zimbabwe. There is no vaccine that is going to come to Zimbabwe, whether it's donation, whether it's been procured by government, that is just going to come through private, and it hasn't hasn't been registered by MCAS. I think we learned also those issues we got with ivermectin, where everybody was going for the dog ivermectin, not even registered, no human ivermectin. All those issues, the lessons learned. And we are really emphasizing that while we want the private sector to come on board, whatever comes to Zimbabwe, government has to take charge, and the Medicines Control Authority of Zimbabwe has to do what it needs to do, what is mandated to do. And this is really under the Emergency Use Authority. Quite clearly, no one ever had COVID-19 vaccine before. It's a new drug, if you like, and hence it is actually in the in terms of the Section 75 for Medicines and Allied Substance Control Authority. And of course, the Pharmacovigilance and Clinical Trials Committee will implement vaccine vigilance plans. Vaccine consignment shall be physically verified and cleared by MCAS, as I've just pointed out, and the consignment shall be cleared on the basis of the standard vaccine lot release documentation. So these are routine standard things that we do. Zimbabwe is really, really well known to have the experts to be able to do that. You are all here right now, a number of you. We just need to continue working together. I do know we have challenges and so on, but I want to urge you that we are just there to continue working together, to hold each other accountable, and to continue following the science that we need to. So this is really, I want to dwell on it, but to simply say, ministry is considering a study to confirm immunogenicity of the product in the local population. These are some of the guidance that have actually come from our Medicines Control Authority of Zimbabwe, and urging that these are some of the conditions that need to be followed properly and clearly. If I come on to the planning, the existing national immunization structures and committees are being strengthened and used to coordinate vaccine deployment under the guidance of Ministry of Health and Childcare. The Zimbabwe National Immunization Technical Advisory Group, the ZIMNITAG, I don't know if Dr. Gona mentioned this, because he is actually the lead on this one, is providing evidence-based recommendations and guidance to facilitate the fully informed government decision-making processes. And quite clearly, they have their science from the WHOs, the SAGE, the what have you, you name it, including some of these conversations that we're having here, where they then also put that science together, that guidance, they give it to government. Of course, it doesn't always say what guidance you provide is all going to be taken on board. There's also that relook and so on. The Interagency Coordinating Committee, ICC, was tasked by government to spearhead planning and preparations for COVID-19 vaccination under the guidance, once again, of Ministry of Health and Childcare. Remember, this is a public health emergency. And the HANS ministry continues to play a very critical role. And of course, myself here working with all those bodies to make sure that we are including everybody, interagency. We have the UNICEF, the WHO, and so on. And the chair of that particular one is actually somebody who is not government, but once again, they're working very closely, guided by Ministry of Health. Public partnership initiatives are being coordinated by Ministry of Finance and Development. We have had so many private sector people coming on board, asking how can they order vaccines for their employees. Interestingly, business seems to be very keen on just getting vaccines for their own people and their spouses and so on. And we are working hard to really make sure that they are appreciating that this is a public health issue. Vaccination is not about just you, the individual. One of the speakers here talked about herd immunity or population immunity, and we're saying, and another one actually mentioned the fact that you are not immune for forever. You might have to be vaccinated every year and so on and so. So those messages going out to the private sector, as they seem to think you just order, you vaccinate your people and you're done, without realizing there's really need to continue collaborating, because we have got to vaccinate the right percentages for us to get this virus out of Zimbabwe. And so Ministry of Finance is doing that to really make sure that they are coming on board in the right way. Ministry of Health will, of course, implement effective deployment of the COVID-19 vaccines through the Zimbabwe Expanded Program on Immunization, the Zimbabwe EPI. We are well known, Zimbabwe, by WHO and others for having a very strong EPI program, and there was definitely no need for us to try and reinvent the wheel. Unfortunately, those who are not in the health sector are not aware of that. And so you see quite a bit of the panic that you're seeing, whether it's from business, from other players, thinking Zimbabwe is not prepared. There is no plan. How are they going to do this? There's no way that Zimbabwe would do such a thing without proper planning, without using the structures that are already in place, strengthening them, working with our supporters, the WHOs, the UNICEF. All that has been happening, is happening, and we're almost there. And so I'm just trying to reassure you there. In terms of resource mobilization, Treasury has set aside $100 million for both procurement and operational costs. The total cost estimated for the budget, Ministry of Health worked on this one, and I think I'm competing with somebody else.
That's fine, it's fine.
Okay, and I agree with the speaker, was it Dr. from UK, that some of our figures there are not quite there. This is just like, come on, let's move with it. The things are happening now, but we are very aware. We're actually revisiting using the WHO costing tool to really make sure we've got realistic costing there. But we're still very happy that our Treasury has set aside that huge amount that should be able to cover us reasonably in terms of procuring the vaccine itself, which will be the largest perhaps cost. But of course, the most important also operational costs that we really clearly need to have. So I won't dwell on those details because we're still really looking at it. Many well-wishers, including the private sector, as I've pointed out, other nations, as well as international and regional funding initiatives such as the AU, the WHO Gavi COVAX facility. I think there was a lot of hype the past 2 weeks or so, people saying Zimbabwe is refusing to sign and so on. But I want to assure you that while there was some delay, there was genuine need to really understand the processes to make sure that we're doing what was right for Zimbabwe, and a lot of advocacy with science from some of you on this platform really obviously demonstrated that when you do it together, you can actually move forward. So we are very much participating in the AU initiative officially, as well as the WHO Gavi facility officially as well. Before, we were participating unofficially by simply using the tools from the COVAX facility, for example, for the assessments that we did in October, November last year, to see how prepared we were, to make sure that when we identified the gaps, we were working on them. So work was actually happening from way back, never mind the signings, the what have you, that some were focusing on. There was no dropping of the ball in terms of just getting ready from the technical side of things. And I'm happy we're almost there. So I won't dwell on that one. Those are the budgets that I was talking. The target populations, really, again, this is really a summary of it. We're going to look at it, but phase one, as I said, stage one, frontline workers, port of entry, personnel, ZIMRA, immigration, you name it. Stage two, village health workers, chronic illnesses, people with that, elderly populations. And then phase two, lecturers, all school staff, population, and other staff at medium risk, depending on the epidemiological picture of the disease as we move forward. And of course, everybody else. So the plan that is there is to try and go through all those. Again, remember we said it's voluntary, and hence we're not saying everybody will be vaccinated. We're really pushing and trying to make sure that everybody appreciates the benefits of us having it as a nation and as an individual, of course, and so that we can really run with, so that at the end of the day, we don't have 90% of the people saying they don't want to be vaccinated. The human resource is a big issue for us in Zimbabwe, and I'm speaking to those who know the challenges on the ground. But just to say, and this is specifically why we're saying we're using existing structures and we're not trying to create new structures. The EPI, the Zimbabwe Expanded Program on Immunization Unit, has been working flat out to try to do the planning, put in place our detailed plan that I'm talking about now. And as we're speaking, yesterday we had the provincial medical directors, other meetings were happening. I met with the UN, the donors, the heads of agencies, while ministry was meeting with the PMDs to really describe and talk about what we're doing, to make sure we're on the same page, to make sure that their questions come on board, their contribution come on board. And I'm happy to say we're really moving as fast as we can. It's almost like development of the vaccine, where in the past it took years, according to one speaker earlier, but this time it took such a short time. So in the past, these kind of processes would take quite a long time. But this time around, they're taking the shortest period of time. As we're speaking, people are being trained. We are already putting in place tools, you name it, everything is actually happening in the same time. And the provincial and district trainers will in turn train healthcare workers at their service deliveries, online, in person, blended learning. All those processes are actually also going to be used. And of course, the areas of storage, communication, surveillance will also be looked at. I won't dwell too much on this. These are really nitty-gritties of the actual delivery itself. Supply chain management, I think, again, we are really looking at the processes that we've always done, but really strengthening them and making sure that distribution follows the channel that we've always used. Central vaccine, we have since in our assessments looked at nut farm and other places, private sector, where we can actually store at the right temperatures and so on. So that is what is happening. I'll skip that one. Others have said, do you have the facilities? Yes, the Gavi, who have supported us with our Zimbabwe expanded program in immunization, the measles, the rubellas, the what have you, have actually also supported us with a lot of equipment and so on. As I spoke with in my high-level coordination meeting with our partners, UN and so on yesterday, we also talk in terms of what are some of the deliverables they can put on the table, what support they can bring on board. Here's our plan. Can we start looking at it? And indeed, there are actually grants that are earmarked from the World Bank and other places, earmarked to support just the logistics of things, and hence perhaps our resource can actually then focus on making sure that we are covering the procurement if we need to. But as I said, there are initiatives that are actually also donating resources and so on. But we do have things in place. So the supply chain, as I've mentioned, distribution planning based on target population, ensure adequate supply of potent vaccine to all eligible populations and so on. I think some of you have already seen some of these slides that came out yesterday. So I want to spend a lot of time because I think it's important for us to discuss. These are some of the things that are already in place that we're looking at. Once again, if you see that that orange sign there, which says Ministry of Health, the Health Development Partners, that was actually supported and funded by one of the initiatives that we're working with our partners and so on. But once again, ministry leading. So we do have things in place. We will have challenges here and there. There's no two ways about it, but move, we shall move. And we are doing just that. And these are other details that perhaps I don't think need to be highlighted here yet, but I think the advocacy, communication, social mobilization, that is information dissemination. Once again, I'm truly grateful for being on this platform, because this is just the beginnings. You need to understand what's happening so that you are actually also disseminating in the right way, so that we really cut off this social media, half informations, misinformation. It's really killing us. And we're actually spending more time on that when we should be spending that time on other important things. So I won't dwell too much on that. And of course, the most important thing is vaccine safety monitoring and management of those side effects when they do occur and so on. And of course, ministries, EPI and the National Pharmacovigilance and Clinical Trials Committee, MCAS, are the main drivers of vaccine safety and surveillance. And we do have tools already that we have used for other vaccines, other medicines, that are again being looked at, strengthened, improved on, guided by WHO and others that we are actually using. So that is also under control. Safety, again, these are additional details. I'm not going to dwell on them at all, because you are aware based on the other, and this is a picture that you are also very familiar with because we have used it many times before for other things. In terms of, my apologies, it's a bit, it's not very focused, but just to remind you that it is in place. Biohazard immunization, waste management, again, it's not something new, so I'm not going to dwell on it. Monitoring and evaluation is very critical. Development of an M&E framework to guide planning and implementation. This is again one of the areas that MCAS specifically said in their guidance and so on. This has got to be in place, and the teams are working flat out to make sure that there is something in place, solid and useful. Pre-vaccination demographic data collection, preparedness assessments. I think I already talked of the two that we already did and we continue doing to make sure we see where we are and so on. So those are some of the details I won't dwell on. So with those few words, I hope it has sort of made us aware of what's happening so that your questions are informed. Thank you. Just finally, just for your information, this is the structure. Many have also asked how can we be part of this coordination? How do we work? This I've showed it in a number of presentations, but I think it's relevant here where we have the interministerial task force at the top. We've got the experts advisory committee. Some of them are on this platform. We've got what we call the working party of permanent secretaries of the ministries that are dealing with the multiple sectors that are concerned with COVID-19, myself being part of that, working with the working party. Yesterday, we had a weekly 2 p.m. meeting to prepare and put reports from the subcommittees that you see at the bottom there to then present to the to the task force on Monday, every Monday, for them to appreciate what's happening, for them to then take to cabinet on the Tuesday, every Tuesday, to then be able to make informed decisions as a government. But I want to remind you that while we recommend, give signs and so on, there are always different views here and there, but the basis of what Zimbabwe is doing, as you heard from cabinet, is really science-based, science-driven, and you are the scientists here that are actually giving us that input. So thank you for that.
Thank you very much, Dr. Mahumbo, for the wonderful and very eloquent presentation. I think let's go into questions so that we don't lose any more time.
Hello.
Hello. Yes. Go ahead. Go ahead, yeah. Eugene.
Yeah. Just one question. I think it's a very simple one, and maybe somebody could help. Now, I see in the statistics there is records of cases, and then there is records of recoveries. I think the last statistic was mentioning something like 35,000 infected people and 30,000 recoveries. Now, I'm just wondering, so, and there is 1,000 deaths, so meaning about 3,000 to 4,000, sort of, I don't know whether to say, that's the difference that's there. How are recoveries being, how do you define recoveries, and how is it being measured in the statistic?
I can answer that now. Yes. Thank you very much. We're guided by WHO. You know, remember WHO at one point was saying recovery is someone who gets a second test negative and a repeat negative. Then you say they have recovered. With the science, again, coming from you, the scientists, they realize, no, you don't need to repeat test anymore. But if after 10 days of not having any signs and symptoms, you don't have anything, you can actually be declared. So to answer your question in terms of how then is that recorded or followed up, the healthcare workers are then supposed to follow up people in the homes and find out whether, based on their records, you were diagnosed on day one and 10 days late, and because you just wanted to travel, but you didn't have any symptoms or anything like that, 10 days later, you still have nothing, you're declared recovered. The challenge I'm sure you're already seeing is the fact that the health personnel are struggling to chase up everyone, and you can't quite call everybody recovered when you haven't seen them, when you haven't heard them. Maybe they went asymptomatic then, and then they became symptomatic, so they cannot be called recovered. So in fact, as we are strengthening our response, one of the recommendations that we've given as part of preparing for a potential or preventing a potential third wave is to really focus on case management, looking at the community management aspect of things. Before, there was tended to really just say, let's focus on the hospital because they're the critical ill, the ones who are isolating, recovering at home, they are fine. They are not. And hence, we are actually adopting the idea of the rapid response teams for surveillance, but having rapid case management teams that really go out into the communities and ensure that those cases are indeed recovering, they record properly, and our recoveries are tallied properly. But this is an area that everybody globally is actually struggling with to reconcile.
Okay, thank you.
Cool. Lots of hands.
Yeah, fine. Yes. I think next on the line is Mfaro. Please go ahead and ask a question.
They must be muted.
Okay. Thank you very much. My question to Dr. Mahompa is: are there any trials currently going on in Zimbabwe to identify which COVID-19 variants are currently circulating, given our case fatality rate, which is higher than everyone else? I think it might actually be prudent if we know which variant we have. Do we have our own variant, or we have the South African one, or the same as everyone else?
Thank you for that very important question. I will first speak like I'm not a scientist at all and say, you know, because of our borders with South Africa, it's good to just assume we've got them here. We cannot pretend because people are just coming in and out and so on. But to then come back to the specific question that you have, yes, we did do genome sequencing of the first wave, and we didn't have any variants at that time. Our people were working with the UK lab. I can't remember its name now, but we then decided, no, this is not a one-off thing. We have to continuously do this, and hence we're really looking again, appealing to some who have the resources for us to have our own equipment for that. I'm happy to say last week we actually received one of the huge machines to do that, and people are now being trained so that we move on and do that. We wanted to send to South Africa, but South Africa is so overwhelmed with their own issues. They just haven't signed the document that is supposed to be signing for the collaboration to do that. And we did that way back, but they quite clearly are focusing on their own. So we are exploring all that, but we're just happy that we now have the equipment and we are moving forward with it to make sure that moving forward, we are able to be indeed guided by science. But in the meantime, reason should also guide us. We're saying our borders are not, we are not like this is Zimbabwe, South Africa is across the oceans and so on. We are the same land, and hence as some of those assumptions are actually guiding what we're doing.
Okay, thank you very much. I think next is on the line is someone on the Galaxy M10. We might have to skip that one. Okay, Fano Mamvura. Do you still have a question, Mamvura?
He's forgotten.
Hello, yes. Anyone who's ready to ask the question? Maybe we have 3 or more questions and then we close down.
Yes, Doc, I have a simple question here. I'm Dr. Mashairi. To Dr. Mahompa, just a simple question. If at all we don't reach the 60% of the population vaccinated, what will happen? People are a bit apprehensive, as it were, now. And although you are saying a lot is taking place in terms of information giving to the people, but a lot of questions are coming out, so much so that we may struggle to get to those figures. What will happen if we don't?
Thank you, Dr. Mashaire. You're absolutely right. That's our concern as well, as I said at the very beginning, and we're saying, but continue to say, nobody's going to be forced to take this. But remember, we said vaccination is our second preventative front. The first one being what we should really be doing. If we're really all doing just that, wearing your face mask properly and so on, that in itself is a vaccination, if you like. It prevents it and so on. So we will continue to strengthen those measures as we continue to get people to appreciate what vaccines can do for them. So it depends on how we really ramp up and step up our communication, advocacy, how you, the scientists, also come on board to explain some of the issues that come with the vaccines, whichever vaccine will be put on the table, so that people are appreciating the need for them to have it, and indeed we move forward. So like you, I really am not sure what will really happen in terms of the science of it, but in terms of what we will do in terms of our response, we're very, very clear. We continue pressing on with the first preventative measures front, while we also on the second measures front, we continue the advocacy to try and get as many people as on board. Remember, we also said we are also putting everything on the table. We're not just guided by one vaccine. We are looking at working very closely with the WHO, some of the vaccines that have been explained here, presented here, which is what and so on. So we're not just saying we have one and one only. Some who might not want one might want the other one. We will see as we move forward.
Okay, thank you. We have questions from Chireka Rukweza and then finally Dr. Chatati.
Dr. Mal.
Yes.
Thank you very much. Thank you very much, the organizers and also the speakers and also Dr. Maomba. My quick question is on, I just need a clarification on vaccination. There's a mentioning of testing of antibodies and swabs. I just want to find out why we are doing that and how you're going to do it. And then if someone is positive or negative, are you going to stop the vaccination? If you could clarify on that, please.
There's a national, we have just completed the national prevalence survey. Our scientists have finished analysis. They're working with ZIMSAT for weighting and so on, so that we have very real clarity in terms of what the situation is in Zimbabwe. Very, very soon we're going to have it. I keep saying that, but unfortunately our scientists are also trying to be very, very careful to make sure that what is issued out is a proper science and not just a quick and dirty process. This actually went through Medical Research Council of Zimbabwe and so on. So we will know. But I hope I'm understanding your question. In terms of someone actually answered earlier to say if you've had the disease and you've recovered, you still should get the vaccine, the vaccine. So that's the approach we're taking based on that science. If you're ill from it, then again, it's you, the scientists out there, who will tell us through those advisory committees and so on, whether one who is still ill from it. Though I don't think if you have measles and you're ill from it, you would be going for a vaccination. Not at all. I think you need to recover first, and then maybe later on you can have it. That's how I would respond to that. But again, I'm eagerly waiting to.
Sorry, Dr. Maomba. My question is on the statement that was produced yesterday. It says blood tests for antibodies and swabs. A sample will be required to determine the potency. That is on desk 2. I don't know what it means. That's why I just wanted checking on clarification.
I'll need to check and see what you're really talking about. But as I said, we're running fast, trying to really keep up, but still be guided by science. If there's any issue that came out that is not quite clear, we'll certainly clarify it.
Thank you very much.
Dr. Male, I'm Galaxy M10.
Oh, okay, please go ahead.
Dr. Mawera.
Yes, Mkoma, go ahead.
Yes, Dr. Maomba, just one quick question. do you have epidemiological data, a profile of who has been affected and who has died to guide your vaccination program? Or we are just adopting from what others have done and applying that profile for vaccination?
Glad you've asked that. If you remember, I said our vaccination program is guided by 12 strategic areas. And I specifically took time with the first one, which I said background, is said the context is critical. And that is part of that context, looking at what is on the ground, what our numbers saying, who has. And yes, we have a line list of all the people who have actually, we are reporting on, sometimes a bit behind, but we are busy updating it to make sure that we're very clear. We have also analyzed some of the deaths that have happened. I know in my last Experts Advisory Committee, we had Prof. Makunike Mutasa coming in with some of the data from the private sector. Their labs are looking at it and so on. So that is the strategic area that I said was the first one, to look at the context, not the usual context. Zimbabwe has a population of that. Some people are going to Mozambique. That context includes our own data that would then help guide us. It's an uphill battle. I will never lie about our challenges at all. There are a few gaps here and there, but we continue to hold on to that, and we continue. This is one of the reasons why I'm also saying I'm so eagerly waiting to see our national prevalence survey, because it will also give us an indication.
So you have bits and pieces of data which you are using at the moment, but not the whole profile of
No. We don't have bits and pieces of data. We've got all our data. We're analyzing it. We're strengthening it. We are continuously updating. I think bits and pieces gives a very interesting social media type soundbite.
Yeah, that's why I'm asking, so that you are clear, because you said they are still analyzing.
We will always be analyzing. Analysis, when you're having an epidemic, you never have globally, Google it up, globally, there's not an epidemic where you have up-to-date data on that day. Hence, you are always still continuously analyzing it, putting together, updating it, because the epidemic is happening.
Yes, thank you.
Thank you.
Okay, thank you. I think we've had enough questions.
Please allow me.
Okay, one last, that will be the last question. Please go ahead. That will be the last question.
Yes, thank you very much.
And me too, please. I have been raising my hand for some time.
I've got a question here for Dr. Mahomva. Thank you. You mentioned a statement where you said the use of RDTs have improved access to testing. In RDTs, I think you imply rapid diagnostic tests. There is no country in the world which uses rapid diagnostic tests, which are antibody-based, for them to determine their numbers. And I'm also looking at your Zimbabwe epidemic curve for confirmed cases as of the 5th of February, 2021. There's a very big spike which would alarm anyone, any scientific person in the world, to say, what is happening in Zimbabwe? Is it the new variant, or it's because we have got a lot of these new RTDs, which are RDTs, which are now being used to confirm cases? And if you look from the statistics or scientific basis of the random diagnostic tests, they've got a 40 to 60% sensitivity and some 30 to 40% false positives. And if these are the basis of our epidemiological judgment and assessment, no wonder why we would have such spike and will get everyone into panic alarm. Can you please explain more about these RDTs? Yes.
Thank you. Please, not if I said RDTs, maybe that misled you, but if you look at my slide carefully, it says antigen. And this is based on the latest WHO guidelines, which actually recommend making a diagnosis using the antigen test kits. antigen, and they have clarity into the guidance in terms of who should be tested using antigen asymptomatic in institutions and so on. So no, we are not using the antibody, not at all. And we're very clear about the science that is supposed to inform our epicurve, our epidemic, and how we respond. Thank you.
Thank you very much. Dr. Tatira, your last question. This is the final one. You're anxious to answer. Yes.
Thank you, Chair. Thank you, Chair. I think in Zimbabwe, we do have two pillars of health delivery, which are the public and the private sector. What role do the coordinator see which the private sector can play in the vaccine rollout?
The private sector needs to come in a bit stronger by ensuring that they are availing their data. Yesterday, I said I was meeting with the UN donors in a high-level coordination meeting. Permanent Secretary acting was meeting with the PMDs, and at that time, there was also issuance of saying the private sector, let's have your numbers in terms of healthcare workers so that they are included. So that's the beginning of it. I also said the Interagency Coordination Committee for Vaccination is, while it's guided by ministry, the chair is not a ministry person. It is coming from the private sector. Hence, the private sector comes through those initiatives. At the very end, I showed you the structure of how we are actually managing this response, where we have subcommittees. Each subcommittee is led by a specific ministry. If I take the labor and social welfare, which is dealing with the social welfare of individuals, they deal with the quarantine facilities, they deal with business, labor, a tripartite, and so on and so on. Last week, they had a big meeting where they then brought in their recommendations last week, but one which were tabled at the task force on Monday. We met yesterday as a working party on Friday at 2, where again some recommendations came in, which we are looking at. One of them actually came from this particular subcommittee, looking and saying, how can our clinics be utilized? And we're saying, okay, it's back to us re-emphasizing that if you are a clinic in Zimbabwe, private clinic, you are supposed, you are a sector that deals with the Ministry of Health and Childcare, and hence that should be the structure, the existing structure that needs to be strengthened so that we are not having parallel structures. I think one of the big challenges that we've seen with this COVID-19 response is the fact that while we opened up for private to try and come on, there's a bit of chaos with everybody thinking they can just do what they like. We've seen some funny clinics being set up, people even threatening medicines control or medicines, medical and dental practitioners councils when they come to inspect them. How dare you come and look at my, it's all those kind of things that we are saying, if we're not careful and we start trying to say we're standing alone as private without strengthening the very good existing structures that Zimbabwe has, we will not do ourselves a good service. And hence, to answer your question clearly, those structures just need to be strengthened. Private sector is part of this initiative.
All right. Thank you very much, Dr. Mahomba. And thank you, everyone. I think we've come to the end of our meeting. I would really want to. Hello.
Afternoon, Dr. Mwale.
Yes.
Thank you. Can I ask my question?
Okay, ask your question and then we stop. Let's stop. I think we have to move on.
Thank you, Dr. Mahomba, for such a nice presentation. My comment is on your planning on sustainability. Given the evidence now that the vaccine will be more of like a flu virus-like vaccination, which may be done annually, if not biannually, I don't know. So do we have any plans more so when we are relying more on donors and also other partners?
But I want to disagree with you completely that we are depending on donors. In my presentation, I very clearly said Treasury has set aside $100 million for this process. But as government, we so appreciate collaboration with other initiatives such as the COVAX, the AU, private sector, individuals, you name it. And I did also say Treasury has been tasked to lead those collaborations as they resource mobilize and so on. So that's what we're doing. At the beginning, I also hopefully clearly indicated that we are making sure this is not a new process. We're including it into the existing processes of the Zimbabwe expanded program in immunization. Yes, that had tended to focus on children, but remember, we also brought up the mums, which is not just vaccinating the children. We brought up the hepatitis and so on. So working with existing structures make it possible for us for that sustainability that you have asked about. That is one of the reasons why we chose to use existing structures to strengthen them so that they are also addressing the other vaccines, but at the same time, bringing this one to be part of as we move forward.
Okay, thank you very much. I think we have come to the end of the meeting. The quality of the presentations has been very high. I think we will probably all accept that it has been the quality of presentations has been very good, but also even the quality of the questions that were asked. So this turned out, in my opinion, to be a very good meeting. So I think with that, we come to the end of the meeting. Hopefully we have some platform where people can ask questions without having to convene a meeting, because obviously there are a lot of questions to be asked. But I think this is safe to allay most of our anxieties, because we are all anxious about the vaccines, naturally. So, but I think a lot of questions have been answered here, and some of the questions have been created, yes. But I think it has been a good meeting. Thank you very much, everyone.
Thank you for having me.
Thank you, Gary, for organizing.
Thank
you. Thank you, guys. This has been a very powerful meeting. I hope we'll have another meeting in the near future.
Thank you.
Thank
you very much.
Thank you, everyone. Excellent presentations.
Thank you. Appreciate you.
Thank you, everyone.
Thank you. Thank you all.
Thank you. Bye-bye.
Thank you all. Bye-bye.
Thank you,
Merci
everyone.
beaucoup.
Atenda chaizvo.
Siyabonga kakhulu.
Thank you.
Thank you.
God, thank you. Bye-bye.
Thank you so much.